Welcome. This is Anna speaking, and we're going to continue working on our senses. So you have those gross anatomy videos that you've probably already watched by now. And now we're going to work on some physiology videos. And this one is going to focus on some basic information on eye physiology. It's not going to go into too much detail, but just enough. All right, you'll remember from earlier when we talked about neurophysiology, we had some special types of cells called bipolar sensory cells. And we're going to start looking at those. But then we also start changing some of the names and some of the, for some of the other cells that are within the pathway. And these are still sensory cells, but we're not necessarily calling them bipolar, even though they technically are. So we've got the classic bipolar, and then we've got ganglion cells. And then over here we talk about horizontal cells and amacrine cells. Okay, so let's first of all remember what you're doing. So over here, whoops, let's change the color. We've got the sun, all right? So this is where all your light energy is at, okay? And then over here, this is the bone of your eyeball, okay? So your eyeball is sitting within that structure. So light is going to come in this direction, so right here is the vitreous humors. It's going to go through this, okay? All the way through this over to here, okay? So with the retina, so we're looking at the structure of the retina now, which you talked about in those anatomy videos. So the light actually has to go past where the axons of cells are, past these cells, past those cells, and get all the way over here to where these bipolar cells, which are called rods and cones, these are our photoreceptors. So these are the two classes of photoreceptors that we use for vision. These are going to be embedded in this tissue right here, okay? So that light is coming all the way over here. Now when this is stimulated, then you can transmit a signal to the bipolar cell here. So you've got the rods and cones, and then you've got the bipolar cell here, and then we can start changing up the names, and we can start talking about the ganglion cells. I've got another slide of this. We'll look at that in a minute. Now I want you to notice that the cones, to me, look like squid, and the rods look like blue whales. Okay, so these are my blue whales, and these are my squid. Okay, and one isn't eating the other, all right, or fighting. So, but they do work together. Or not really work together, but they're both there, and that helps me keep them straight. I don't know why the whale thing does, but the other thing that I missed was right here. We've got the label on the amacrine cell, which is coming over to right here, okay? And these are all part of the neural pathway, and I've got my horizontal cells coming in right here, okay? But this is my classic bipolar cell. This is called the ganglion cell. And this is a rod, let's change the color, rod and a cone, okay? And basically, I've got to get the light here, and then it's gonna go here, and then it's gonna go here, and then it's gonna go here, and then it can go up to the brain, okay? Let's look at this from a different perspective. All right, so this is just another drawing of the exact same thing. But sometimes it's nice to see it twice. So over here, this is where the light is going to come in, okay? And then over here is the bone, okay? And so light is going to come in here, and it's not going to do anything until it gets to here. When it hits these cells, that's when you can start picking up the sensation, and then it's going to go from here to here to here. And sometimes you've got, like, say, this one, and then this is 1B, and then it might be doing a serial pathway there, and then you've got 2, and you might have a 2C over here, and then you've got 3. And then these are the axons of the optic nerve. Okay, let's move on. All right, I'm sure I mentioned this in the gross anatomy videos, but there was a summary slide that talked about rods versus cones. And so you need to memorize this. I mean, just flat out memorize it. So rods, light, all right, actually, I don't like the way it transferred the PDF. So this is cones right here, and we're just gonna black that out. We're just gonna black that out, okay? And then this is cones over here. And it looks better on the PowerPoint when you download it. So cones are gonna do bright light, all right? And then somewhere over here I've got color, okay? So cones are color vision and it functions in bright light. Rods are shades of gray, all right? Shades of gray, right here, monochromatic vision, shades of gray. And it's gonna be in dim light, okay? This one also has to deal with dark adaptation, which we're going to talk about in a little while, whereas cones are not. They adapt really quickly. I've got the location and the fovea centralis for cones, and then all spread around the retina for rods, okay? Let's see what's next. Let's look at what light is. All right, this should not be new, all right? You should have seen this in high school, and you've seen it in other classes and so forth, okay? So what is light? So when we're talking about vision, we're detecting visible light energy. And that is just a very narrow part of the spectrum right here. And then you can see right here where it's blown up, and you've got your blues, all right, your greens, your yellows, orange, red, and so forth, okay? Now, when light travels, it's basically kind of a wave going in a straight line, and when it hits a refractive surface, so something that is flat and it can bounce off of, it's going to bounce at an equal and opposite direction, so equal and opposite angle, okay? This is important because of the shape of our eyeball, the cornea, so the shape of the cornea and the lens. So right here it's telling you what happens if you have a concave lens, all right, so it's like this, then the beams spread out when they come in, all right, they're going to go like this and they're going to spread wide. Whereas if you have a convex, it takes lots of light and it gets it to go to a single point over here where it kind of makes it converge, okay? Let's go to a Blackboard slide, and we're going to talk about light transduction, or excuse me, not light transduction, the refractive apparatus next. Okay, so this is my note slide, and I guess I'm not going to go to a Blackboard. I'm just going to put this up here. You need to memorize cornea, aqueous humor, pupil, lens, vitreous humor. You've already done this in the anatomy video lecture, so I already went over this. With the cornea, you want to remember that it is convex. So this is the shape of that, excuse me, why am I drawing the lens? So you want to remember your cornea is this shape, okay? And so that light its job is to take light and converge it so that it's going to go all towards a single point in the back, okay? All right, let's look at how that lens works next. All right, so here we're looking at a convex lens, and we're looking at the way it's going to converge light. So I've got light energy over here, and I've got a beam that's going this way, and a beam that's going this way, and a beam that's going this way. My lens is shaped like this, so that when light energy hits this spot, it bounces in equal and opposite direction and takes it over here. This one, because it's hitting it on fairly straight, it just goes straight through and goes over to here. This one comes over, it's going to hit this side right here, bounce at an equal and opposite direction, and it's going to come over here and hit that spot. Okay, so the purpose of the lens is to take light energy from lots of different areas and all get it over here to a single focal point, okay? Now, one of the things that's interesting with the way our lens works, and you've probably seen this with mirrors, is that the light actually inverts the image. So it's reversed and upside down. And that's what we're showing you on the bottom. So over here, we've got yellow on the left, and then over here, you've got, okay, so that's lower yellow left, and then up here, upper yellow right, okay? And then over here, we've got lower purple right, and then up here, we've got upper purple left, okay? So it's reversed and upside down. All right, let's talk about how we're going to focus next. All right, my notes slide, memorize it. Distant vision versus near vision. So distant vision is what our eyes are actually adapted to. Theoretically, if your eyeball is the right shape, you don't have to focus your lens to see. It's going to see. So the ciliary muscles are completely relaxed. The lens is stretched as thin as it can get. All right, and you can just see far away. In order for the human eyeball to see near vision, you need to do lens accommodation, okay? And that's to see anything that's closer than 6 meters. So we've got 3 little reflexes that we use for this. All right, ciliary muscles are going to contract. So this is parasympathetic stimulation. All right, and the tension is released on the suspensory ligament. Lens recoils and bulges, which reduces the focal length so that you can see something closer, okay? Now, one of the things that's interesting, and this kind of goes with this right here, this should be a bullet point underneath here, is that as we age, the proteins in our lens thicken. And as they thicken, it becomes much harder for them to bulge and unbulge. And basically what happens is with age, your ability to focus on things that are close to your eyes is reduced because of this thickened lens that can't accommodate very well anymore, okay? So this is the first thing that you need to do is lens accommodation. The next thing is you've got pupil constriction, or the inverse, which is pupil dilation. And the reason you're doing this is you want to control the amount of light coming into your eyeball in order to get the best accommodation possible. So if you need to do close vision, often you need more light than you would if you were doing far vision. So you're going to dilate. Unless you're in a really bright place, in which case you're going to constrict so that you don't blind yourself, okay? And then finally, you've got eyeball convergence. So you had accommodation, and then you have eyeball convergence. And this is where, because we have binocular vision, that means humans have good depth perception with having our eyeballs in the front and by coordinating our 2 eyes to work together. So we basically are going to use the eyes and try to look at the same thing at the same time, and we're going to try and get the light all to go over to the fovea centralis. That allows us to have this binocular vision where we can perceive depth. We can see where the next branch is on the adjacent tree in order to jump to it, okay? Whereas if you have your eyeballs on either side of your head, that becomes very difficult. I was reading a book recently, a fantasy book, and it had these creatures who were like animals that were turned into humanoid shaped, and the hero was trying to teach them to shoot guns. And what he found was that the animals that had the eyeballs on the front, so like they came from gorillas and wolves and stuff like that, let's just not even think about the whole hand thing. They had the depth perception to be able to aim a gun and shoot a bullet. But the humanoids that came from horses, because their eyeballs were on either side of their head, they just totally couldn't shoot anything accurately at all because they had no depth perception, because they could not get their eyeballs to converge because their eyeballs were on either side of your head. So when you're writing a fantasy novel and you're making up alien creatures and you want them to shoot guns, make sure their eyeballs are both in front. Otherwise, their aim is going to suck. On the other hand, if you don't want them to shoot all the humans, maybe you make them so that their eyeballs are on either side. We'll see. All right, here's a classic example of an eyeball. Look at how pretty this is. So a nice, relaxed, round eyeball the lens, the ciliary muscles are relaxed, which causes the suspensory ligaments to pull, and that causes the lens to flatten out, okay? That means when I have light here and here, they're gonna come over here, hit, go equal opposite direction, come over this way, go in this way, hit the lens, equal in opposite direction, come over here. I've got my inverted image. And it's all gonna come right over here to where I have my fovea centralis, which is important for being able to see something clearly, okay? Now let's look at the next one where we're gonna do near vision. All right, now here we are looking at a nice normal eyeball, but the letter R is really close to the eyeball now. So what you're gonna do is you're going to contract the ciliary muscles, which is gonna release the suspensory ligaments. I know it's a little counterintuitive, so just memorize it. And that's gonna cause the lens to bulge into this shape. So remember the far vision one, the lens was more flat and thin like that. Okay, so you can see what the shape difference is. Okay, now because this is all bulgy, I can take the light here and we're gonna do this, and we're gonna do this, and what it's gonna do is when it hits the lens equal in this opposite direction, it's gonna go right here and it's gonna go right here, so that this is focused in on the fovea centralis region. Okay, so that you have clear vision. All right? All right, let's talk about when things go wrong. I have no idea why this orange line was imported when I imported this. It's really annoying. Myopia, hyperopia, astigmatism, and presbyopia. Okay, so myopic means you are narrow or near, okay? So people who are myopic are nearsighted, which means they can see things close up. What causes this? So this should be the normal shape of your eyeball, okay? So this is normal, all right? Myopic, the eyeball is more like this. Okay, that's a bit extreme, but yeah, all right? Hyperopia, all right, hyper is like a lot, all right? It's an intensity, so these people have better far vision, so they are far-sighted. So you can see far away really well, but not so close, all right? Now, this one, the eyeball, is too short, so it's more like that in shape, okay? Astigmatism. So astigmatism, you remember you got a lens, and then we can also draw an eyeball right here, okay? And then I'm going to change the color, and I'm going to give the cornea right here, okay? An astigmatism is when there is a wrinkle or scratch in either the lens or the cornea, creating an irregularity to the curvature, okay? And then presbyopia, old eye. So I was talking about that lens when it gets old, gets thick, and it doesn't change its shape very well anymore. Okay, and that's when you have to start getting cheater glasses. Okay? All right, let's look at some pictures of these. All right, I am never gonna ask you to tell me what that word is. Who cares, right? So this is your normal eyeball, obviously. I want you to notice the light coming in, hits the lens, all goes right here, all goes right here. The goal is to get it to the fovea centralis for clear vision. Fovea centralis is where you have your highest concentration of cones, as we learned in the gross anatomy videos. Now, for nearsighted, remember, I said that for normal, it's round. For people who are myopic or nearsighted, it's more like this. And people who are hyperopic or farsighted, it's more like that, okay? So here, when the light comes in and it hits this lens, the lens is fine, the cornea is fine, but notice that it's focusing and it's hitting this point. But because the eyeball is way back here, my fovea my fovea centralis is right there. So now the light spreads out before it hits the fovea centralis, so it is going to be blurred. Okay. Down here with the far-sighted one, same thing. Light's coming in normal. It's refracting normal, but it's coming over here. My fovea centralis is actually right there. So when the light is hitting the fovea centralis, it is also spread out and blurred. Okay, and then here it shows you what kind of glasses you get, the shape to the lens in order to fix that problem. Okay. All right, let's look at a few other disorders real quickly. All right, depending on who you end up having for pathophysiology, you'll look at some of these eye disorders. So strabismus, all right, two eyes do not match one another, okay? Nystagmus, rapid uncontrollable eye movement, okay? The two that probably most of y'all are familiar with and probably most concerned with are glaucoma and cataracts because they affect so many people, okay? Glaucoma is where that canal of Schlemm isn't draining the aqueous humors, and then you get buildup of fluids, and that puts pressure on the eyeball, and it could eventually lead to blindness. Now, cataracts are going to be clouding of the lens like frosted glass, all right? The lens gets harder, thicker, and it just doesn't work well. Obviously, if you're looking through frosted glass, you can't see clearly, okay? But nowadays, surgery is awesome. It wasn't so awesome in the past. A lot of people died because they tried to get cataract surgeries that like totally went bad. Like J.S. Bach died from complications of a botched cataract surgery. He was a famous classical musician, if you haven't heard of him. But nowadays it works really slick, and they basically do one eye at a time. They cut out your lens, they put in a fake lens, and you can then see clearly out of that lens. Now, because it's no longer hooked up to the suspensory ligaments, you can no longer do near and far vision with the same lens. So you will have to choose what you want. Most people choose to keep far vision and then use reader glasses for close-up. Although sometimes people choose the opposite. I don't know why. Okay. All right, color blindness is probably one more that you are familiar with because we like to use it in biology classes for years and years and years, to the point where everybody's sick of it, because it's a really nice example for a sex-linked chromosomal disorder, okay, or chromosomal difference. Basically, there are several different kinds of color blindness. The one that you're most familiar with because of the way we teach biology is red-green color blindness, where you do not have the cones to distinguish red and green colors. So most people have 150 can see 150 shades of color, whereas people with red-green color blindness can only see about 25 colors, okay? Now, in case you're wondering, am I colorblind? This is the colorblind test. If you do not see the number 7, you might be colorblind, or this test might be awful. We'll see. You can look into it. All right, let's move on. All right, so looking at color chemistry and how you actually perceive different shades of light, we need to first do a couple of definitions. So first of all, you've got retinol, okay? Retinol is a compound that absorbs light, okay? We're gonna make this compound from vitamin A, okay? Then we have another compound called opsin, or opsins for plural, okay? These are proteins that are going to bind with retinol, okay? And they're gonna bind to make 4 types of pigments, okay? So when we perceive light, we are perceiving the destruction of pigments. So this is really important, okay? So what are our 4 visual pigments, okay? So first of all, let's talk about color vision. So with our cones, we get color vision. So we're going to have 3 types of cones: blue cones, green cones, and red cones, okay? And each absorbs a different part of visual light, okay? So what do these pigments consist of? All right, so blue is going to be the blue opsin plus retinol. Okay. Green is going to be the green opsin plus retinol. And then red is going to be the red opsin plus retinol. So we're combining these proteins. Now when we talk about them, we usually just say red, green, and blue. Okay? Now, in addition to that, so let me see. I'm going to put this little star right here. I'm going to come down here and do another one. We also have what is called rhodopsin. Okay? And rhodopsin is for your black, white, gray, your monochromatic vision that's all associated with rods, okay? And that's going to be a mix of a different type of retinol with the opsin that does that, okay? Now, we always recycle the opsins and the retinols. All right, so we're breaking them apart and putting them back together, then breaking them apart and then putting them back together. All right, let's just look at that real quickly. We'll look at that wavelength chart again. All right, this is that wavelength chart again. So we're looking at, you got blue over here, you've got green over here, and then over here is the red. So the blue reflects blue, the blue cones. The green reflects the green cones, okay? And so you're basically reflecting those part of the light wavelengths, okay? And the opsin is going to vary either between the rods or the 3 types of cones, and that determines what part of the light wavelengths is going to be absorbed versus reflected. All right, let's look at a chart for dark adaptation next. All right, how quickly you can see in light and dark depends on how quickly you can recycle and reassemble the pigments, okay? What you see with cones over here is, so this is showing you minutes in the dark versus retinal sensitivity, okay? Cones recycle their pigments really quickly and reform them. As soon as you've got light, the cones begin to work, okay? In a dark environment, you can see that the cones, their pigments are all ready to go, but they're really not doing anything, okay? In contrast, rods up here, so here you're seeing how much sensitivity you have to the light. The rods are really slow to recycle and reassemble their pigment proteins. So it can take up to 10 minutes, 20 minutes, 30 minutes, 40 minutes for the rods to have maximal recycling and reassembling of their rhodopsin proteins so that they have the most sensitivity to the amount of light that is present, okay? And that's what this chart is trying to show you. Now, if you turn the lights back on, these get overwhelmed, and that's when you like cover your eyes and groan and you're like, oh, too much light. All right, they get overwhelmed. But these go back to functionality immediately. They're very, very fast when you have high light environments, okay? And that's all I'm explaining in these little note slides. All right, let's look at light transduction next. All right, if you remember from before in neurophysiology, we talked about what transduction is. And transduction is when you turn the information from a stimulus into a potential, okay? So that's all we're talking about with light transduction. This is where you turn a stimulus into a local or action potential. All right, you've transduced it, okay? So we are going to do the process of light transduction when light hits the pigment, okay? When light hits the pigment's retinal and opsin are going to separate, okay? This separation, the separation is what triggers change in membrane potential. And remember, change in membrane potential is what creates a local potential or action potential, okay? In this case, light, the way our cones and rods work are a little wonky, all right? They're a little, not wonky, counterintuitive, okay? So when this happens, it actually turns off depolarization of the rods and cones, okay? So it turns off the depolarization of the photoreceptors so that no action potentials are moving from the photoreceptor to the bipolar cells, okay? So let's continue on with this pathway of counterintuitiveness. So in the dark, all right, so in the dark, what you've got going on is the photoreceptors are constantly open. All right, they're constantly open and constantly stimulating their adjacent bipolar cell, okay? By doing this, they're going to release lots of glutamate. And remember, glutamate is a neurotransmitter that is inhibitory, all right? Therefore, glutamate inhibits the bipolar cell. By inhibiting the bipolar cell, then there is no stimulating force of the third cell in the relay, in this case, the ganglionic cell. All right, so it's basically going to not stimulate the ganglionic cell, okay? Now, in the light, all right, what's going to happen is the light energy, all right, it's going to hit those pigments and cause them to separate, all right? They separate. Then, all right, what's going to happen is the sodium gates are going to shut. All right, right? And the receptor begins repolarization. And then finally hyperpolarization, okay? When that happens, you are no longer sending that glutamate out and inhibiting the bipolar cell, okay? Therefore, the ganglionic cell is then triggered to send an action potential, okay? So it's kind of counterintuitive, but technically when light hits our photoreceptors, it turns the photoreceptor off. So the absence of our photoreceptors sending out neurotransmitters is what causes us to see, which is really kind of weird when you think about it. All right, so let's kind of draw this out. So we've got in the dark, all right, it is dark out, okay? So we're going to secrete inhibitory glutamate, okay? That means the bipolar cells. So right here, we're dealing with our cones and rods here. Cones and rods are secreting inhibitory glutamate. So bipolar cells are inhibited, okay? Which means no stimulus to the ganglionic cells. Okay? Now, in the light, all right, and again, we're still dealing with rods and cones, all right, there is no stimulus to the bipolar cells, okay? All right, and this is because light breaks those pigments, okay? So the bipolar cells send their neurotransmitters out, all right, to the ganglionic cell. And then the ganglionic cell is going to talk to optic nerve axons and send a signal to the brain, okay? Because the ganglionic cell depolarized. All right, I hope that makes sense, okay? So for visual integration to work, you got a lot of different things going on, but basically these cells are all kind of working together for it to work. But it isn't one cell to one bipolar cell, so one cone or rod to one bipolar cell to one ganglionic cell. It is actually going to be, so let's say we have rod, cone, rod. God, everybody is sending me messages. And then we're going to go to like one bipolar cell right here. It's a B. And then we're going to have another set of rods, cones, rods, cones, cones. They're going to go to a different bipolar cell, and then those are going to go to a ganglionic cell. And in between, we are going to be borrowing help from horizontal cells and amacrine cells. So that it is creating what we learned in neurophysiology with those various parallel processing pathways. And you don't need to get too bogged down in that. I'm just trying to mention that they are there, okay? All of this is going to work together to enhance and control and finesse our ability to see different parts of the pathways. let's just really quickly follow up one last time with a picture of the retina. All right, so this was that last picture that we looked at this before, and this is going to be actually our second to last picture. So when we got the sun over here, light comes over here. So what we're showing again is blue whales and squids. These, if they stop communicating here, then the bipolar cells will then communicate with the ganglionic cells, and this will depolarize. So we've got rods and cones, bipolar cells, ganglionic cells, but we also have horizontal cells and amacrine cells, which are going to create these parallel processing networks. And then also notice that I don't necessarily have one-to-one cells. So like right here, there are 3 rods going to this one bipolar cell. There are 2 bipolar cells communicating with the ganglionic cell. So again, parallel processing is the way the game is played. All right, let's look at one last picture where we're going to look at the visual pathway with the optic nerves, and then we'll stop. All right, we're going to look at the visual pathways now. And we're just going to make this simple, and we're going to assume that this is a superior to inferior view, so that this is basically your right eyeball, and this is your left eyeball, and this is the right side of your visual field, and this is your left side of the visual field. Often in pathophysiology and other textbooks, it's actually reversed when they show it, but it's very difficult, I think, for people to think of it that way. So we're just going to go with this. All right, now I want you to think about what your light is doing. So we've got light over here on the right, and this light all goes in this direction, okay? So the light from the right side of the visual field hits the left side of the left eyeball and the left side of the right eyeball. The left side of the visual field, colored in blue, hits the right side of the right eyeball and the right side of the left eyeball, so that the left eyeball is doing the left half of is doing the left half of both the left visual field and the right visual field, and the right eyeball is doing the right half of the right visual field and the right half of the left visual field. And you can see that where that's being picked up with the photoreceptors here and here, and they've got it color-coded so that you can see the optic nerve here and the optic nerve here. And what is interesting about this is that you can determine where someone has brain damage based on where they might have blind spots. So if I cut this right here, okay, where this is where my optic nerve is, the person would be blind for the right side of the right visual field and the right side of the left visual field, but they would still see the left side here and the left side here. All right, that's where their brain would be interpreting the information. Okay, but if I cut, like if I had a pituitary tumor right here and it's compressing this area, what you would have is people who can see this part and this part, but they can't see this part and this part. So they get blind spots in a different area because they cannot interpret the inside of the visual field. They can only interpret the outside of the visual field, or vice versa, okay? So I think that's just very interesting the way this comes up. And this is why that optic chiasma is so important for you to learn when you're doing your bony features, is because the eyeballs switch half of their fibers at the optic chiasma, and then the optic tract is going to be carrying all of the right visual field information to the left side of the brain, and all of the left side of the visual field to the to the left side of the brain versus the right side of the brain. Anyway, I'm reversing my left and right as I talk, but I think you're getting the idea. Hopefully I haven't confused you. Anyway, this is interesting stuff. But we're going to go ahead and stop the eye physiology lecture because it's gotten long enough, and you can go on to ear physiology.